Good morning to all of you. So, that day when we finished, we were doing the PBR 322, the vectors. We were doing artificial vectors. We were doing this, and in this, what we did was PBR 322. Yesterday, I sent you the notes also, and it had this PBR 322, and that also showed how many recognition sites it has, the number of recognition sites. Recognition sites are the points where the endonucleases can cut. They can make the cut. These are those points where the endonucleases can cut. So there were 8 recognition sites, and then there was in PBR 322, there was ROP, protein of replication. It was also there. 1, ORI was also there, origin of replication 1, and selectable markers we had. Selectable markers. And there were 2 selectable markers, ampicillin resistance and tetracycline resistance, showing these 2. So this we did yesterday. And then I told you one more artificial vector, which is PUC. This is prepared by University of California. Plasmid, University of California. And it uses enzyme producing gene as selectable marker. It uses enzyme producing gene as selectable marker. Then I told you about TI plasmid. TI plasmid, this is the DNA, or TI plasmid we call it, or TI DNA also we can call it. This is present in Agrobacterium tumefaciens. ME. This is a bacteria which infects the dicot plant. This bacteria infects dicot plants, and when it infects dicot plants, it sends. Suppose this is a dicot plant which is infected by this bacteria. So what it does, that it sends its TI-DNA inside. That means this DNA which is sent inside is a tumor-inducing DNA. This is sent inside. So TI is tumor-inducing DNA. This tumor-inducing DNA is sent inside, and when this tDNA goes into a cell, in that cell, when it is injected into a cell, in that cell it reaches the nucleus, and then this cell multiplies, keeps on multiplying and keeps on multiplying, increase in number, and results in the formation of tumor. That means this DNA has the ability to go inside the cell and then reach the nucleus and help the cell to form multiple copies. So this particular thing the scientists noticed, and they use this particular property of Agrobacterium tumefaciens in their work. how to use this agrobacterium tumefaciens, how to use this agrobacterium tumefaciens, its TIDNA, for sending our gene of interest. Suppose we want to make multiple copies of our desired gene. So what the scientists did to this TIDNA, if this is TIDNA, this is TIDNA. In the TIDNA, they included our gene of interest also. That means to this TIDNA, our gene of interest was also added. So you can see that to the TIDNA, our gene of interest is also added. And when this bacterium, this was inside this gene was this TIDNA, which is now carrying our gene of interest. This is now TIDNA, which has our gene of interest. When this TIDNA, which was present inside agrobacterium tumefaciens, when it was allowed to infect the cell, what will happen? Of course, our gene will also enter the cell, and then it is its property to form tumors. It is a property of TIDNA. That is why the name tumor inducing DNA, because it induces the formation of tumors. Now what happens when this TIDNA, which is carrying now our gene, when it entered inside, it helps the cell to multiply many times. That means our gene also has formed multiple copies. Suppose this is a gene for say uh, disease resistance, so multiple copies of that gene is formed, and then finally what the scientists did, then they extracted that which particular protein which was formed by this gene, that was extracted by the people or by the researchers. So multiple copies of that particular gene has been made. So this TIDNA, which is tumor inducing DNA, has been used for transferring our gene of interest and making multiple copies of it. So another artificial vector which is used is this TIDNA, or tumor inducing DNA, which is used to transfer our gene of interest and to make multiple copies of it. So children, this is understood to all of you? If this is not understood, just let me know, I'll repeat it. Yes, beta. Yes, bache. Yes, bache. Is it clear? I want messages from you. I want message from you, beta. Is it clear? Yes, I'm getting the message that it is clear. So this is another vector which is okay, Vanithi. Yes. Okay. Yes, it is clear now. So this is how the till now we have done PBR322, which is used as vector, then PUC, which is used as vector, TIDNA of agrobacterium tumefaciens, which is used as vector. Now one more is that this takes place in case of plants. This TIDNA, it can infect only the plant cell. So we want something, the same experiment, the desired gene has to be sent to animals also. What to do if we have an animal tissue? Then what can we use in case of animal tissues? Because this TIDNA, which is obtained from agrobacterium tumefaciens, has the ability to infect only dicot plants. That means only in the plants only we can send a desired gene. We cannot do it in animal case, in the case of animal tissues. So what if we have animal tissues? Now there are bacteriophages. Bacteriophages, these are the viruses which infect bacteria. And how does it infect bacteria? It sends DNA inside. It sends its genetic material inside, whether DNA or RNA. Material inside the bacteria. This is how it infects. Now, this can also be done, because in this case also what is happening, that a virus is attacking, this is a bacteria, and to this bacteria a virus is attacking, and this virus, when it attacks, it sends its genetic material inside. So the genetic material of this virus, it comes in, and inside what happens, it uses the machinery of the host and makes multiple copies. Then a head is formed. Okay, this way multiple copies are made, and these are then released out so that they can infect more. This is what happens when a bacteriophage infects a bacteria. So that means, see, multiple copies are made. The same gene has made multiple copies. We can say this gene to make, see, obviously this is going to happen whenever a gene enters, it undergoes the process of central dogma, prepares its proteins, for which information it carries. Okay, whatever information is here, whatever information this is carrying, it will make its own protein, and it will also increase in number. See, multiple copies are also made, and the protein it has to synthesize, it will also synthesize that particular protein based on the information which is present on the DNA. Now, the bacteriophage, when it infects it, what if we send our gene of interest along with its DNA? The virus, the bacteriophage, doesn't know that it will be carrying some desired gene or not. What will happen that if we include our desired gene here in the viral DNA, that can also enter, and that can also enter the host, and there it can synthesize the protein as well as it can form multiple copies. That means multiple copies can be formed, and that particular protein can also be synthesized. So what was done by the researcher, the same thing to this DNA, this our gene of interest was added, and then this virus doesn't know that it is carrying a gene of interest also. So when it injected the genetic material inside the host, what entered along with the bacteria, along with the viral DNA, our gene also entered in. And then using the machinery of the host, by using the machinery of the host, the multiple copies were made, and the protein was also synthesized. Multiple copies were made. The protein which this the inform for which the information it was carrying, that protein was also synthesized, and multiple copies were also made. And this is what bacteriophages does. That means we are using the bacteriophage for our purpose, for sending the desired gene in the host. Here also we use the tumor inducing DNA for transferring the desired gene into the host and multiplying in number, so that it can multiply in number and produce lot of that kind. So here also we used it for our benefit. Here also we have used it our benefit. This particular process is known as disarming. This particular process is known as disarming. We have removed the arm, whatever the arm of this particular bacteriophage, or here this agrobacterium tumefaciens, and we have used these two organisms for our purpose, for transferring the desired gene into the host. So beta, this bacteriophage can also be used as a vector, and the two bacteriophages which are commonly used as vectors, their names are, the most common bacteriophages which are used as vectors are lambda bacteriophage, this bacteriophage is used for the purpose of transferring a desired gene. Along with this, one more bacteriophage which is used for the purpose of transferring the desired gene, and that is M13. And besides this, there are retroviruses which can be used for transferring a desired gene. When we were doing ADA deficiency, in that case, we used retrovirus through which we send a desired gene for making ADA. Those were allowed to infect the lymphocytes so that the lymphocytes can produce ADA. So in that case, if you remember ADA deficiency, SCID disease, in that case, retrovirus was used. So retroviruses can be used, lambda bacteriophages can be used, and M13 bacteriophage can be used to transfer our gene of interest. So till now, I have told you for few. The first artificially cloned vector was PBR322, which was formed by Bolivar and Rodriguez in the year 1977, and that was first artificially made vector. Then we can use, there was another PUC, then we can use TI, tumor inducing DNA of agrobacterium tumefaciens. Besides this, various bacteriophages can also be used, like lambda bacteriophage, M13, and retroviruses can be used to transfer a desired gene or the gene of interest so that it synthesizes the protein based on the information present on the DNA, as well as it can make multiple copies of this. So this is what, anything in this which you could not understand or you want me to repeat? Yes, beta? Anything in this which you want me to repeat? Then I'll tell you screening and about the antibiotic selectable markers. I have to tell you then. I'll start with selectable markers now. Anything you want me to repeat in this? Yes, bache? Yes, I'm not getting any response. I'm not getting any response. Paras Singh, clear, ma'am. Okay. One response I've got. Yes. Any more? Okay, clear. To everybody it is clear. So now, see, in simple language, I don't know how many students are there today. Okay, 16 students. Now, beta. So you have understood what is disarming. Disarming of vectors. That means we can use them, the organisms which harm, for our benefit. We can send the desired gene with them. If it is a tumor inducing gene, it will not produce tumor. Of course, it will inject, because this is its habit to infect the dicot plant cells. It will infect the dicot plant cell, but what will go in with along with it is our desired gene. So these are some vectors. Now with us, in simple language, I just want to tell you whatever the process we did just now. See, basically what happens, this process is used to make, to transfer our desired gene into the host so that the host can prepare the protein based on the information which is present on DNA, as well as it can make, good quantity of it. Now, see, suppose there is a person, and the person, there are not one person, many person who are diabetic, who cannot produce insulin in them. So insulin is provided externally. Insulin is provided in the form of injections. Now suppose, from where that insulin is obtained, for that, what is done? Suppose this is a normal insulin producing gene. we have this gene which is a normal insulin producing gene. We have obtained it from a normal person who can produce insulin normally. Now what is done? This insulin producing gene has to we can use this insulin producing gene to produce insulin for us, so that we can sell it to the people who are suffering from diabetes. Now how can we get lot of insulin which can be provided to the diabetics? So first of all, this insulin producing gene through some vector needs to enter a host. So we will include this insulin producing gene say in a vector which is PBR322. Right now I'm not telling where I have, which endonuclease I have used, and where the cut has been made. I'm not telling you anything like this. This I'll take as a separate topic after this. Now suppose we have taken this insulin producing gene in a plasmid. This is a vector. This insulin producing gene has been taken in a vector. Now this vector will take this insulin producing gene into the host, and let's say the host is E. coli. Mostly E. coli is used as the host, the bacterium Escherichia coli. So this vector moves in this. Now this is the plasmid. Inside what happens? This will increase in number. This will multiply, as well as the bacterial cell will also multiply. This will, the E. coli, they increase in number. So they will increase in number, they will multiply, reproduction will take place, and as a result of that, the bacterial cell will multiply. So what we have got? From one, we have got so many insulin producing genes. From one, we have got so many insulin producing genes. See, desired gene has increased in number. Now this is its nature, because this is insulin producing gene. So inside the E. coli, this will undergo central dogma. That means DNA is there, desired gene. This is DNA. This desired gene is DNA. DNA, RNA, transcription, RNA, translation, protein. Now which protein will be formed here? Insulin will be formed here, and after that, this all the Escherichia coli are collected, they are processed, and insulin is extracted from this and can be sold in the market. The whole process is this. What is biotechnological process? The whole process is this. Okay, now where we have inserted our gene of interest, that vector which is carrying our gene of interest, we have inserted that here. See, all of them, they have our gene of interest, and some of the bacteria, they might not have our gene of interest, they have their own plasmid. See, these two, they do not have our gene of interest. You can see this one, which I have named as A and B. A and B do not have our gene of interest, while C, D, E, F, G, and H all have a gene of interest, all have the desired gene in them. These two, A and B, they are known as non-transformants. These are known as non-transformants because they have not changed. Whereas C, D, E, F, G, H, which have a desired gene in them, they are known as transformants. They are known as transformants because they are carrying a desired gene in them, and these are non-transformants. They do not have a gene in them. So, these are microorganisms. How we will come to know that this particular bacteria has a gene in it, or this particular bacteria does not have a gene in it? Because, bacteria grow anywhere and in any number. So it is very difficult to separate transformants from non-transformants. So for that, the selectable markers are there. This is what I told you. When you go for outing, you wear a maroon blazer, so you can be, so that you can be identified from the normal people who are there. So same way, to separate, to identify transformants from non-transformants, there are selectable markers. And those selectable markers are present in these vectors. In these vectors, those selectable markers are present. Now, how do they help to separate transformants from non-transformants? This is what I'm going to tell you. Till now, is it clear? Are you clear with what are transformants and what are non-transformants? Is it clear, children? It should be very clear to you, because if you do not understand it here, further you will not be able to understand. This is biotechnology, you know something. Okay, Paras Singh is saying yes. I want all from everybody. Even if one child could not understand it properly, just let me know. I'll repeat the whole process. I do not have any problem in repeating. Just let me know that you did not understand. Because if you do not understand it here, you will not be able to understand the whole process. Okay. Sukh Sahaj is also saying yes. Any other? Shall I repeat it? Amit Katoch, are you there? Amit, Mubashir, Karthik? Yes. Yes. Simran, Aditi? Yes, beta? It will be difficult to understand if you do not understand what are transformants and what are non-transformants. Chitra? No response. I'm not getting any response from anybody. Yes, bache? With Vanshika? Vanshika? Okay. so transformants are those hosts which can be E. coli or any other bacteria. Okay. Are those hosts which carry a gene of interest. And these are those which do not carry a gene of interest, are non-transformants. Now, how to separate them? How to know, how to identify that this is transformant and this is not? For that, selectable markers are present. And what are those selectable markers? Now, beta, let's take example of PBR322, which is a vector. So this is PBR322. That day I drew it. I'm drawing it again, but I'll not draw it full, just little. I would like to show you only the selectable markers. So PBR322 was like this. One more ring was here because the thickness was shown. And in this PBR322, we had here ampicillin resistant marker here. We had this ampicillin resistant marker, selectable marker, and we had one more marker here on this side, which was tetracycline resistant marker. So here I have made with the green color. This is tetracycline resistance. Okay, this is one marker, and the red one which I have made is ampicillin resistant. So these are the two markers. What do we mean by resistance? When I say ampicillin and tetracycline, ampicillin is one antibiotic, tetracycline is another antibiotic, and both are very strong antibiotics and are used for treatment for killing the bacteria. Okay. Now, that means ampicillin also kills, ampicillin is an antibiotic. Kills bacteria. Tetracycline is also an antibiotic which kills bacteria. And when we say that this particular plasmid is a part of bacteria, this is a artificially made plasmid and it is a part of bacteria, that means this is a vector, and this vector or plasmid has to be inserted or introduced in a bacteria, which is going to carry our gene of interest. And now I'll draw our gene of interest by this red mark. Here we have ori. This is ori, and near ori we have our gene of interest, which I have made like this. This is our gene of interest near ori. Now what happens? Now, when we say ampicillin resistance means wherever this vector is present, that bacteria, that means if we introduce this vector into a bacteria and then we sprinkle ampicillin on it, that bacteria will die or not? See, this is introduced in a bacteria. This vector is introduced in a bacteria. This is a bacteria in which this vector is introduced. Now, when we sprinkle ampicillin on it, or when we grow this bacteria in a medium with ampicillin, will this bacteria die or survive? See, it has got ampicillin resistance. It will survive, yes. Why will it survive? Because it can withstand ampicillin. It has got ampicillin resistance. It has got resistance for ampicillin. So if this vector is in a bacteria and this bacteria is picked up and grown in a medium with ampicillin, it will not die. Normally what happens? Ampicillin kills bacteria. But here, because this has a marker with ampicillin resistance, which has ampicillin resistance, so this bacteria has got now this bacteria has got resistance for ampicillin, and if it is grown in a medium with ampicillin, it will not die, it will survive. Same way, if this bacteria with this plasmid, and the plasmid has a marker for tetracycline resistance, if this is grown in a medium with tetracycline is another antibiotic which kills bacteria. So if this is grown in a medium with tetracycline, or some tetracycline is sprinkled over it, will this bacteria die or survive? This has got tetracycline resistance also. So if this bacteria now grows in a medium with tetracycline, or some tetracycline is sprinkled over this bacteria, it will die or survive? It will survive, because it has got resistance for tetracycline also. It can resist tetracycline, it can resist ampicillin. That means if this bacteria, if this plasmid which is carrying a desired gene is introduced in a bacteria, inserted in a bacteria, and then this bacteria is a transformant. Now this bacteria is a transformant because it has got the vector with a desired gene. We can call this bacteria transformant. Because this now has got a desired gene. If this bacteria is grown in a culture of ampicillin, it will survive. We have got a number of bacteria. I'll make it here. I'll just make it here. This is a plate. This is a petri dish with ampicillin in it. This is ampicillin. Ampicillin antibiotic. Now if we grow the bacteria here, so out of these bacteria, some are transformants and some are not. So out of these, when it is grown in a culture of ampicillin, which is going to survive? This will survive, this will survive, this will survive, this will survive, because they have this plasmid which has ampicillin resistance, whereas this will die and this will die, because they do not have ampicillin resistance. Same way, if we take another culture, and in that culture we take tetracycline, and in this tetracycline also the transformants, the bacteria are grown, and we don't know that there are we are not sure whether, we can never be sure whether the vector has been introduced in the bacteria or not, because it is such a method that you cannot come to know whether the plasmid has entered the host or not. Now this is Another type of culture which has tetracycline in it, tetracycline, antibiotic in it, which kills the bacteria. Now, out of all, some of them are transformants. Say these three are transformants. Now, what will happen? When it is grown in a medium with tetracycline, the transformants will survive, whereas non-transformants will die. So we'll be able to separate the transformants from non-transformants. So this is one of the method by which these selectable markers can be used to identify the transformants from non-transform, to separate them, rather to separate the transformants from non-transformants, and then we can pick up the transformants and culture them for producing our gene, for producing the protein from the information which is given in the gene. Now, this is one thing. Now, there is a term, but I think the time is over. I think I should leave the meeting. The time is over.